Shortly after he was born, baby KJ Muldoon seemed perfectly healthy, but within days he became lethargic and stopped feeding, signs that led doctors to a devastating diagnosis, an ultra-rare metabolic condition called CPS1 deficiency. The disease is a mutation that breaks the liver's ability to handle protein. The protein turns into toxic ammonia. That ammonia travels upward, flooding the brain, causing rapid swelling, neurological damage, and eventually a coma. Under standard care, KJ's life was defined by extreme fragility. While waiting for a liver transplant, his survival depended on a near-zero protein diet. Even a minor cold or a common stomach bug could stress his body enough to trigger total organ failure. The medical playbook offered no immediate fix. To save him, his doctors had to create a specific targeted intervention before his body encountered the first everyday stressor. A collaborative team at Penn Medicine and the Children's Hospital of Philadelphia proposed a radical intervention, building a custom genetic cure in record time. While this type of genetic engineering typically takes a decade of development, the researchers only had a 6-month window before KJ's condition reached a breaking point. To maintain scientific objectivity, the lab team enacted a strict blinding protocol. They shielded themselves from knowing the baby's name or seeing his face, referring to their subject only as Patient Ada. They abandoned the hunt for a mass-produced medicine. Instead, they entered a hyper-focused blind race to engineer a genetic correction for exactly one human being. First-generation CRISPR wasn't an option. Its molecular scissors cut the DNA double-strand, sometimes leading to frayed, unpredictable repairs. Instead, they used base editing. These quality editors reach into the DNA and smoothly swap a single nucleotide, changing an A to a G, without ever breaking the strand itself. This required microscopic precision. The tool scanned 3 billion letters of KJ's genome to find and rewrite the single flawed block. The lab entered a furious sprint. They mapped the mutation, customized the editor, and successfully tested the mechanism in humanized mouse models. By the end of the window, the team had built a microscopic tool designed exclusively to rewrite the singular biological typo threatening patient Ada. Engineering the tool in a test tube was a milestone, but it created a new logistical hurdle. They had to transport the machinery directly into the liver without triggering an immune response that could be just as lethal as the disease itself. They packaged the base editor inside a lipid nanoparticle, a microscopic fatty sphere. This is the same technology used in mRNA vaccines, allowing the cure to slip past the body's defenses and enter the liver cells undetected. With the delivery system ready, the researchers petitioned federal regulators for emergency approval. This was an N-of-1 request, a plea to use an unproven, custom-built therapy on a single human life. Authorization was granted. At 6 months old, KJ received the tailored dose that would define the rest of his life. On February 25th, the lab work finally became a medical procedure. KJ received the first of 3 infusions, starting with a cautious low dose to ensure his system wouldn't reject the nanoparticles. The clinical team watched the blood work for weeks, looking for a signal in the noise. Eventually, the data shifted. His toxic ammonia levels dropped into a safe range, and for the first time, his liver could process a normal amount of protein. Months later, KJ faced the ultimate stress test. He contracted a common cold and a gastrointestinal bug. In the past, these would have triggered a fatal crisis. Now, his system neutralized the viruses and he recovered with no complications. KJ is the first known person to receive a fully personalized, in vivo CRISPR base editing therapy, a treatment developed and executed in just 6 months. The success creates a new complexity for medicine. Scaling this requires massive financial and logistical resources, and it is unclear how the healthcare system can provide these bespoke therapies to every child with a rare disease. But the biological landscape has shifted. Mass-produced drugs are no longer the only option. KJ is living proof that we now possess the technology to engineer a genetic cure for a single individual.